Hello everyone, I am Dr. Tejendra Kumar. I am going to teach you methods in biostatistics. Unit number 6, Statistical Tests. Today we are going to the, learn about chi-square test, which is the part of this statistical test unit. Let's start with the introduction. The chi-square test is a statistical tool used to compare observed results with expected outcomes and evaluate the likelihood that any differences are due to chance. In biology, researchers often need to determine whether observed data fits expected patterns, or this is especially common in genetics, ecology, and evolutionary biology. So the chi-square test is a statistical tool which is uh, used or to evaluate whether there is a significant difference between the observed frequency and the frequencies we would expect under a specific hypothesis. Okay, or we can say that it is one of the simplest yet most powerful tools used to test hypotheses about categorical data, and it is especially used in the many of the branches in biology, as I told you, like genetics, ecology, behavior, and epidemiology, etc. Now, types of chi-square tests. The chi-square test compares observed data with data we would expect to obtain according to a specific hypothesis. It answers the questions, likewise, are the differences between observed and expected values due to chance, or are they statistically significant? So, the types of chi-square tests uh, have two types. Uh, First one is goodness of fit test, and second is test of independence test, or which is also called as contingency table analysis. Goodness of fit test used to determine if a sample matches a population with a known distribution, whereas the test of independence or contingency table analysis determines if two categorical variables are related, for example, genotype versus phenotype, etc. Now, formula. As you can see here, chi-square test, uh, chi-square is equal to sum of bracket observed minus expected whole square divided by expected. So where O means observed frequency, E means expected frequency, and the sum is taken over all the categories. Next formula for degree of freedom, DF, is the for goodness of fit test, degree of freedom is calculated by number of categories minus 1, or we can say n minus 1. For test of independence or contingency table, we can use the formula as rows minus 1 multiply with columns minus 1. Now let's see the assumptions of the chi-square test. Assumptions means these are the some conditions that where we can apply the chi-square test by seeing the data, or by just checking the values. We can assume that okay, in this case we can apply the chi-square test. So before performing the chi-square test, it's important to ensure that the data meets certain assumptions. The first one is observations must be independent. Means each observation should be independent of others. For instance, each plant in a genetic cross should be considered independently, or each individual's choice in a behavioral experiment should not influence another. Next, expected frequency in each category should be generally be more than or equal to 5, because this is also called as adequate sample size. The expected frequency for each category should generally be at least 5. If any category has fewer than 5 expected observations, the chi-square test may be inaccurate, and alternatives like Fisher's exact test should be used. Next, data must be in frequency or count for, not in percentage or means. Categories should be mutually exclusive and collectively exhaustive. Here is the important point that categories or categorical data. Chi-square tests are determined for categorical data. For example, data grouped into categories like tall versus dwarf or infected versus non-infected, no, but not apply for continuous data. Mutually exclusive means each observation must fall into exactly one category. We can see in the tables, like there are all the rows are following all the columns, or all the data which are showing in a specific row may represent at least one of that column, and each row is following at least one of the uh, and vice versa, each column is following the row, and similarly for the columns. Okay, so in that way we can say mutually exclusive and collectively exhaustive, because there is a grand total. Interpreting the chi-square values. Supposedly we have calculated the complete uh, thing, and now we are trying to interpret that how we could write the results. Then this could be uh, the most important thing, because it is giving you summary. So here is a chi-square distribution table. Always compare with the chi-square distribution table using a appropriate degree of freedom. Supposedly you have calculated some things, some values, and now you are trying to interpret on the basis of the given values or calculated values. So there are the some rules. Okay, how to check the table? Let's see this one. Okay, here are the few values. Okay, likewise you can see here in the given table you can see some values. So on the upper row there is a number which is given, that is the significance level alpha. You can see here 0.99, 0.975, 0.95, 0.9, 0.1, 0.05, 0.025, and 0.01. So these are the significance level which is called as alpha. But on the first column we can see here there is a degree of freedom, so which is given at the leftmost column, that is 1 2 3 4 5 6 and so on up to 1000. So by seeing this table you can calculate the values. Supposedly we want to we want to check a value on the chi-square table, so always check the values at the significance level 0.05. Okay, so here it is given, sorry for the some technical errors. because it is repeating the animations again and again. Let's see again. So here is the 0.05 alpha significance level. Now this is the column you have to check for all the values at the level of significance at 0.05. It means 95% confidence is there and the significance level or alpha level is 5%. Now supposedly you want to check the value for degree of freedom 1. So let's check this row. So before the 1 or in the row of 1 at the intersect of 0.05 you can see the value 3.84. So here is a value and here is the final value which is 3.841. So now interpret the results. If critical value means table value is greater than the calculated chi-square, which we will calculate in the table, we will calculate in our question. So it means P is greater than or equal to 0.05 level. So in this case, we will fail to reject the null hypothesis. Alternatively, we generally call it as accept the null hypothesis, but the correct term is fail to reject the null hypothesis. We generally not use the word accept. So the conclusion is fail to reject the null hypothesis. Okay, now see, the P is the probability that the difference is due to chance. If critical value is less than the chi-square, it means P is less than 0.05. In that case, we will reject the null hypothesis. There are some limitations to apply the chi-square test. First, cannot be used for small sample size. As we discussed earlier, that in each categories, expected frequency should be more than or equal to 5. Okay, so that's why there is a one limitation. Does not indicate strength or direction of the association. So this is also a limitation. We can generally see the numbers, but direction and strength we can't interpret. Not valid if expected frequencies are too low. As I told you, 5 is the adequate at least number. Last one is this test is unsuitable for continuous data, as we discussed earlier also. For example, height or weight, as it only works with categorical data. Let's take some examples. This is the first example where we will apply the goodness of fit test, and the topic is taken from the some applications of the chi-square test in different fields of biology. So first example is taken from the genetics or Mendelian genetics. In the Mendelian genetics, we can apply goodness of fit test. Let's take an example. Problem is, or question is, in a pea plant cross, you expect a 3:1 ratio of round means dominant to wrinkled means recessive seeds. You plant 160 seeds and observe that round are 120 and wrinkled are 40. So let's calculate and get the answer. So step 1, calculate the expected frequency. So do total of all the given observed values, like round 120 plus wrinkled 40 means 160. So expected values are always calculated on the if a ratio is given, then like 3:1 ratio. So total means 3 plus 1 is equal to 4. So Round, as we discussed, this is the dominant and we expect it as 3 out of 4. So that's why 3 by 4 into 160 is equal to 120. So expected ratio for round is 120. Similarly, expected frequency for wrinkled is 1 by 4 into 160 is equal to 40. In second step, we will compute the chi-square value. This is the formula for the chi-square value. Chi-square is equal to sum of bracket observed minus expected whole square bracket upon expected. Let's put all the values in this formula. Chi-square is equal to 120 minus 120 whole square upon 120 plus 40 minus 40 whole square upon 40 is equal to, so there is a no difference and we will finally get the answer 0. So chi-square value, or we can say that calculated chi-square value is 0. Let's determine in the next step or third step, determine the degree of freedom. So df is number of categories minus 1. Number of categories are always calculated as, for the goodness of fit test, n minus 1. So n, the number of categories. Here are two categories. One is wrinkled, second is round. So n minus 1 means 2 minus 1 is equal to 1. Step 4. Interpretation or conclusion. A chi-square value of 0 means there is perfect agreement between observed and expected. This supports the hypothesis that the observed segregation fits a 3:1 Mendelian ratio and there is no deviation or no difference is found. Example number 2. Goodness of fit test. It is also applicable for Mendelian genetics. I took one more example of the monohybrid cross. Let's take a problem, and that is if a cross between heterozygous tall means capital T small t pea plants and you expect a 3:1 ratio of tall to dwarf, you observe tall 72 and observe 28. Now there is some difference and calculate the chi. So the answer will be taken as, first step, calculate the expected value. So total is 72 plus 28 is equal to 100. Expected tall are 3 by 4 into 100 is equal to 75 and expected dwarf is equal to 1 by 4 into 100 is equal to 25. But you can see the tall and dwarf are different than the expected. Let's compute the chi-square value in the second step. So here is the formula which is similar as above or previous formula. So chi-square is equal to 72 minus 75 whole square upon 75 plus 28 minus 25 whole square upon 25 is equal to 0.48. Let's determine the degree of freedom in next step. Number of categories minus 1 is equal to 2 minus 1 is equal to 1. Let's interpret the conclusion. Critical value at P is equal to 0.05 is 3.84, which is checked from the table. Since 0.48 is equal to 3.84, we fail to reject the null hypothesis and results fit a 3:1 ratio. The result is likely due to random chance. So if there is a little difference, then we can say interpretation or conclusion in this way. Now take next example, goodness of fit test for a sex ratio in a population. In a sample of 200 newborn turtles, sex was determined as males 110, females 90. Does it differ significantly from 1:1 sex ratio? So we calculate the expected values as 1 by 2 into 200 is equal to 100. Females 1 by 2 into 200 is equal to 100. Why we took here 1 by 2? Because ratio is given as 1:1. It means similar for both the genders, male and female. So if we add 1 plus 1, it means 2. So 1 by 2 into 200. 200 is the n number. So in this way, we expect 100 males and 100 females. Let's compute the chi-square value. Similar formula we will apply, and chi-square is equal to 110 minus 100 whole square divided by 100 plus 90 minus 100 whole square divided by 100 is equal to 100 by 100 plus 100 by 100 is equal to 2. Let's determine the degree of freedom in next step, which is 1, because 2 minus 1 is equal to 1. Finally, interpretation or conclusion. Using a chi-square table, the critical value at P is equal to 0.05 and degree of freedom 1 is 3.84. Since 2 is less than 3.84, that's why we fail to reject the null hypothesis, and or we can say that it is just accepted, and there is no significant deviation from 1:1 ratio. Means it is a value that can be accepted easily because there is no large difference or no significant differences there. Question number 4. Goodness of fit test for the gene linkage in Drosophila. Here is a problem. You cross two Drosophila and observe the following offspring. Parental type 400, winged red eyes, and 390 wingless and white eyes. Another two are recombinants. These are 100 winged white eyes and 110 wingless red eyes. You expect 1:1:1:1 ratio if the genes are not linked. So calculate the expected value, because there are 4, 1 plus 1 plus 1 plus 1. So expected frequency per phenotype is equal to 1000 divided by 100. 1000 divided by 4, or we can say that 1 by 4 into 1000, that is similar, and it will give the result 250. Means we expect 250 from all the types, whether both of the parental types or both of the recombinant types. We expect 250 from each, but our numbers are different: 400, 390, 100 and 110. So it means there is a difference. Let's compute the chi-square. Similar formula will be applied. Chi-square is equal to 400 minus 250 whole square divided by 250 plus 390 minus 250 whole square divided by 250 plus 100 minus 250 whole square divided by 250 plus 110 minus 250 divided by 250 whole square. Because 250 is the expected ratio, so O minus E whole square divided by E. So that's why we are taking the 250 from all the numbers. Okay, so chi-square is equal to, in the final conclusion we can see here the values are 336.8. Let's determine the degree of freedom, that is 4 minus 1, because there are 4 categories, 1:1:1:1 ratio. That's why 4 categories. So 4 minus 1 is equal to 3. Interpretation or conclusion. Critical chi-square value at P is equal to 0.05 and degree of freedom 3 is 7.815. Since 336.8 is greater than, or we can say that highly greater than 7.815, but the value, calculated value was 336.8, which is very high. The deviation is highly significant, and this suggests the genes are linked because the difference is very high, and that's why we reject the null hypothesis, and we can say that this is or deviation is highly significant. Highly significant difference is there, and that's why we can say that the genes, suggested genes are, yes, clearly linked. In our null hypothesis, we said that the genes are not linked, but yes, now we can say that null hypothesis is rejected and we can say that genes are linked. In the next question, goodness of fit test applied for behavioral ecology or animal activity preferences. Now here is the problem. You study the activity patterns of 200 animals in an experiment with two treatments, day and night. You count the number of animals active under each treatment and find. In the day, 120 animals are found active, and in the night, 80 animals are found active. Your hypothesis is that the animals are equally likely to be active during both the days and night. Equally, it means we expect 1:1 ratio. So, calculate the expected values. If the animals are equally likely to be active during both periods, we would expect 100 active animals in each period because the total number is 200 as given in the question. Now, compute the chi-square value. So, chi-square will be calculated from the similar formula: 120 minus 100 whole square divided by 100 plus 80 minus 100 upon 100. Sorry, 80 minus 100 whole square upon 100. So finally we will get a calculated chi-square value as 8. Next, determine the degree of freedom in the next step. So df is equal to 2 minus 1 because there are two categories, day and night. Now n minus 1 is equal to 1, or we can say that 2 minus 1 is equal to 1. Now match the table value and we will reach up to this conclusion or interpretation. Critical chi-square value at p is equal to 0.05 and degree of freedom is equal to 1. So the critical value for these p and df will be 3.84. Since the calculated chi-square value is 8, whereas the critical value, which is taken from the table, that is the 3.84, so we reject the null hypothesis, indicating that there is a significant difference in activity patterns between the day and night because the calculated value is high. In that case, we will reject the null hypothesis, and if we reject, then we cannot say that they are following the similar patterns. And that's why we can say that there is a significant difference in activity pattern. There is a significant difference, means there is a large difference in between the 8 and 3.84, and that's why the we can say that there is a significant difference in activity patterns between the day and night, and both are following the different time zones. Like some animals are following the day and some animals are following the night, and both are different and both are unequal. Okay. Question number 6. Goodness of fit test applied for the behavioral ecology in the food preference. Supposedly, you test 4 food types on fruit flies. The number of flies choosing each is apple 22, banana 26, orange 28, and pear 24. Now calculate the expected value. Because we are expecting the equal number for all the fruits, that means we are expecting that all fruit flies should go for the all fruits equally. So if flies have no preference, expected equal number should be 25 per fruit, because the total is 22 plus 26 plus 28 and 24 means 100, and that's why we are dividing by the number of fruits, means 4. So 100 divided by 4 means 25 per fruit. So compute chi-square value. Formula is same, and chi-square is equal to observed 22 minus expected 25 whole square divided by expected 25 plus 26 minus 25 whole square divided by 25. Similarly, plus 28 minus 25 whole square divided by 25 plus 24 minus 25 whole square divided by 25. So we will get the final result. Chi-square calculated value is 0.8. Let's determine the degree of freedom. Because there are 4 categories: apple, banana, orange and pear. So 4 minus 1 is equal to 3. Interpretation or conclusion. Critical chi-square value means table value at P or significance level 0.05 and degree of freedom 3 is 7.815. We can see it, or we can check it from the chi-square distribution table. Since 0.8 is less than 7.815, therefore we can say that there is no significant difference is detected for all the food types, and it means all the fruit flies are almost equally approaching for all the fruits, because there is a no more difference. 22, 28 or 26, we can say that they are around 25. So only 1 or 2 or highest 3 differences given there, and that's why there is no such preference for the there is no such preference for the any fruit, and that's why we can say that all the fruit flies are choosing fruits uh, without any preference. Now, example number 7, test of independence, which is applicable for habitat and species presence. So problem is, do two bird species prefer different habitats? Let's take this example. Species A live in forest number are 30 and in grassland 10, whereas 20 individuals of B species are living in forest and 40 in grassland. Okay, calculate the expected value. So because this is the test of independence type of chi-square test, and that's why the expected frequency is calculating by the different formula, and that is row total into column total divided by grand total. Okay, so these are the final calculated value. You can see here in the table I have written all the values. So row totals you can see here 40, 60, and in the column total you can see here 50, 50, and grand total is 100. Okay, so for species A in forest, here you will get the 40 from the total row total of A in the forest, and column A forest is equal to 50. So 40 into 50 divided by 100 is equal to 20. For species A in grassland, here you can see A, then again 40, and in the column A grassland 50. So 40 into 50 is equal to 200. Divided by 100 is equal to 20. Similarly, for species B in forest, 60 into 50, because row total 60 into column total 50 divided by 100, which is grand total. Similarly, for species B in grassland, so row total 60 into column total 50 divided by 100 is equal to 30. So expected frequencies for A forest 20, A grassland 20, A B forest 30, and B in grassland 30. Now compute the chi-square values. Formula is same, and the chi-square is 30 minus 20. 30 is the observed value, which is given here, A forest. So 30 minus 20 is the expected value, whole square divided by expected value, which is 20 again, plus 10 minus 20 whole square plus divided by 20 plus. Similarly, for the B, 20 minus 30 whole square divided by 30 plus 40 minus 30 whole square by 30. So we will get the final result of the chi-square value as 16.66. Let's determine the degree of freedom. So here is the different formula for determining the degree of freedom in the test of independence type of chi-square test. So the formula is rows minus 1 into columns minus 1 is equal to 2 minus 1 into 2 minus 1 is equal to 1 into 1 is equal to 1. Let's interpret or have the conclusion. Critical value at P is equal to 0.05 and degree of freedom 1 is 3.84. Since 16.66, which is the calculated value, is higher than 3.84, means the difference is statistically significant. We reject the null hypothesis, and bird species do differ in habitat preference. And we can see here a species is likely to be live in forest more as compared to the grassland, whereas species B want to live in grassland and more as compared to in forest. So there is a significant difference we can observe here. Next example is for the test of independence in case of fish and their mutation. So problem is a population of 500 fish is examined for the presence of two mutations, and are these mutations independent of each other? Now this is a very tricky question, and here only one value is given, that is the total number n, and second, which is just told you that two mutations are there. Now you have to assume all the values yourself. In this way, in these type of questions, or in this type of question, you always have to assume the values, and that's why I have taken this example, so that you can understand that which type of questions can be asked. Step 1, define the hypothesis. So null hypothesis is saying, or H naught is saying, the presence of mutation A is independent of mutation B. We should always take as asked in the question. So that is the null hypothesis. Secondly, alternative hypothesis, which is H1 or H alpha. The presence of mutation A is not independent of mutation B, or we can say that dependent, right? Now calculate the expected values. We need a contingency table showing the number of fish with combinations of mutation A and mutation B. Since the data wasn't provided explicitly, so let's assume a typical data set. So I have written here a table, and I have just assumed the number from my own mind. You can write any of the number. Okay, so mutation A present, mutation B present in both the columns and rows. Now I have taken the values like this way: mutation A present, whereas mutation B also present. It means that value is 150. Mutation A present and mutation B is absent. Here is the value 50. Mutation A present, mutation B mutation A absent, mutation B present 100, and mutation A absent and mutation B present, that is 200. Now calculate all the values. Now here you can see row total and column total. So the numbers are given here. Row total 200, 300, and column total 250 and 250. So grand total is 500. Now see the complete expected frequencies also. 200 into 250 divided by 500 is equal to 100. That is the expected frequency for A present and B present. Similarly, A present, B absent. 200 into 250 divided by 500 is equal to 100. Next, for mutation A absent and mutation B present, here 300 into 250 divided by 500 is equal to 150, and mutation A absent, mutation B absent. Here 300 into 250 divided by 500 is equal to 150. So in this way we have found that mutation A present here, the expected frequencies are 100 and 100, and in case of A absent, 150 150. Compute now compute chi-square values. Formula is same and place all the values. So observed frequency is 150 minus expected frequency 100 divided by expected frequency 100. In this way we have written all the values here, and we will get the complete chi-square value as 83.34. Let's determine the degree of freedom. Rows minus 1 into columns minus 1 is equal to 2 minus 1 into 2 minus 1 is equal to 1. Now interpret or give the conclusion. Critical chi-square value means stable value at alpha 0.05 and degree of freedom 1 is 3.84. Since 83.34 is very higher than or too higher than 3.84, that's why we can say that we reject the null hypothesis, and there is significant association between the mutation A and mutation B, because question was asked as independent of each other. But here, because we are rejecting the null hypothesis, that's why we will take the opposite answer, and that is we will say that yes, there is a significant association, not independent. So significant association between mutation A and mutation B. Therefore, they are not independent, means we can say that they are dependent. Now take one more question, that is the question number 9. So it is applied for the test of independence for the right hand and left hand. A study examines left versus right-handedness in males and females. Is hand preference independent of sex? So define the hypothesis. Null hypothesis H0 is hand preference is independent of sex, whereas alternative hypothesis H1 is hand preference is not independent of sex, or we can say that hand preference is dependent of sex, or it means we are saying that means supposedly some persons are right-handed and some persons are left-handed, so it is not related to the gender in case of null hypothesis, and in case of alternative hypothesis we are saying that yes, if there is a male, then it will be more chance to be right-handed or left-handed in this way. So we are just assuming the two hypotheses, null hypothesis and alternative hypothesis. Now calculate the expected values. We need a contingency table, and let's assume the typical data set, because it is also not given in this question as well. So I have taken this table. So right-handed males 100, left-handed males 30, right-handed females 120, and left-handed females 20. So calculate all the values. Here you can see row total, column total, and given the frequencies, expected frequencies also. Okay. Now compute the chi-square values from the same formula, and you will get the result of chi-square as 3.462. Determine the degree of freedom. Rows minus 1 into columns minus 1. So 2 minus 1 into 2 minus 1 is equal to 1. And interpret or conclusion. So chi-square values at alpha is equal to 0.05 and degree of freedom 1 is 3.84. Since 3.462 is less than 3.84 and we fail to reject the null hypothesis, means there is, or we can say that we are generally accepting the concept of the given question or null hypothesis. So there is no statistically significant association between the sex and hand preference in the sample, as asked in the question that they are independent of sex. So hand preference appears to be independent of sex. now this is the last question of this exercise. Okay, set test of independence for the deer and parasitic habitat. Let's take 50 deer in a habitat A and 50 in habitat B are examined for parasites. A have 30 infected individuals and B have 10 infected individuals. Is there a significant is there a difference in infection rates? Let's define the hypothesis. Null hypothesis is saying that infection rate is independent of the habitat, and the alternative hypothesis is saying that infection rate is depends on habitat. So calculate the expected values. We need a contingency table. Let's assume the typical data for as we did for the previous questions as well. So these values, and now calculate all the expected frequency, compute the chi-square value, and final value is 16.66. Determine the degree of freedom, which is 1, and now come to the interpretation or conclusion. So chi-square values at alpha is equal to 0.05 and degree of freedom is 1 is 3.84. So calculated table chi-square value or critical chi-square value is 3.84. Since 16.66 is I have written the wrong thing here. It is greater than 3.84 and we reject the null hypothesis because it is higher, and there is a statistical significant difference in the infection rates between the habitat A and habitat B. Okay, let's correct this thing. It is higher. Okay. Okay, kindly excuse me for the noises of aeroplanes. Okay, so critical chi-square value at alpha is equal to 0.05 and degree of freedom 1 is 3.84. Since 16.66 is higher than 3.84, that's why we reject the null hypothesis, and we can say that there is a statistically significant difference in infection rates between habitat A and habitat B. Deer in habitat A have a significantly higher infection rates, which we can see here, than those in habitat B. Similarly, infection in the habitat B is lesser than the habitat. In habitat A have lesser infection as compared to the habitat A, means habitat A have higher infection rates. So there is a significant difference which is found here. So this is about the chi-square test. If you have any query, then please write me and comment here. And if you found any ambiguity or any error in the complete video, then please do write me for the correction. I'll be more than happy, and I appreciate your efforts and suggestions. Thank you so much.